wonderful PU2 champions out there. I hope all my dear children are doing absolutely great. Well, today I am going to discuss the answers for the previous year question paper for your midterm examination. So children mostly find enough resources, okay, for your final boards, but they do not know what to refer to for the midterm examination, and here is one such video which is definitely going to help you in some ways for your midterm examination. I would want to say always, but obviously you would not believe me, so let's give it some ways. And my dear children, make sure you're listening to me carefully, not going anywhere else. Okay, so I'll try to make this video as short and concise as possible. I have already written down the answers, I just have to discuss it with you guys. So stay over here, listen to me carefully, that's your challenge. Okay, let's not get deviated by anything out there, and if at all at any point you think that, oh my god, this video is definitely helping me out, do not forget to hit the subscribe button, do not think of anything else as well. Okay, let's go ahead then with the first question out there, and that is, in a monocot embryo, shoot apex and the leaf primordia are enclosed in a hollow foliar structure called, we discussed this in the live. So this is a monocot embryo. The first thing that you need to remember is that now they're talking about shoot apex and the leaf primordia. Leaf primordia means basically the baby leaves out there which will give rise to the big leaves later on. So shoot apex and the leaf primordia, my dear children, over here is covered by something that is known as coleoptile. Well, you can call this region as plumule as well. Why? Because this is the region that gives rise to the entire shoot, that is the stem, the leaves, everything out there, and it is covered by coleoptile. So you can remember P in plumule is there, and over here in coleoptile also there is a P. Now, if you're asked about coleorhiza, where is it? So radicle, which is the future root, my dear children, below that you have coleorhiza. It has an R in it, and radicle also has an R in it. This is exclusive for monocot embryo. It's not found in dicot embryo. Okay? I hope you have got something out of it. This picture is also very important, by the way. Now going ahead with the next question, question number 2. A student squeezed an orange seed while doing an experimental procedure and was astonished to see so many embryos in it. So within a seed of the orange, you find so many embryos. What do you call this condition? You call this condition as polyembryony. That means multiple embryos. Polyembryony, it is found in citrus fruits such as orange as well as mango. Now my dear children, do you think fertilization has taken place over here? Fertilization means what? Male gamete, female gamete coming together to form the zygote. Here, without fertilization, zygote has been formed. Where? Inside what? Inside the ovule, my dear children. So everything is happening within the ovule. And you know what? Anyway, zygote is being formed inside the ovule. Now you will not call this structure as ovule, instead you will call this as seed. And this zygote will transform into what? This zygote slowly transforms into what? Embryo. That's how it grows up. Now over here, suppose there are multiple embryos coming inside and forming here in the seed. You call this condition as polyembryony. And you do not see fertilization to happen over here. So this is also an example of what? Apomixis. So formation of seed without fertilization is known as apomixis. It can happen in different ways. One such way is shown over here, where what is happening, you know, within the ovule. If the ovule is like this, and here is the place where the zygote, where the egg, the female gamete is there. Okay, here is the place where everything is going to happen. The zygote formation, embryo formation, everything is going to happen. The cells around this is known as nucellus. Okay, it is known as nucellus. Some cells, they get inside this structure, and once they get inside this structure and divide, my dear children, it is going to give rise to the embryo. Once it is inside, anything that comes inside, my dear children, can become an embryo. You can think like that, it does not actually happen like that. So now, here suppose multiple embryos are being formed, you call this structure as polyembryony. You call this, procedure as what? Polyembryony. And who is making these embryos? The cells of the nucellus. Now, ma'am, is fertilization happening over here? No, which is why this is also an example of apomixis. Okay, now, products of syngamy. Those embryos were products of syngamy. Syngamy means what? Fusion of male and female gametes. Do you think it's the product of that? No. Haploids. Haploid means half the number of chromosomes would be there. But over here, my dear children, polyembryony usually happens when the nuclear cells, as I said, come inside and form the embryos. The nuclear cells, each cells are diploid in nature. They are just normal cells of the plant. So they would be 2N, diploid, not haploid structure. Please remember that. So this, in the case of polyembryony, it is going to be diploid. Okay, fine. So this is not haploid. Of different size and shape, exactly, yes, because as you can see over here, they are of different size and shape and developed from the secondary nucleus. What do you mean by secondary nucleus? So the two polar nuclei which is there. in the female gametophyte. Okay, there are two polar nuclei which is a part of the central cell, my dear children. These are known as the secondary nucleus. So it is not developed from the secondary nucleus, so that is also not your answer. Your only answer is this thing. This question is a little bit twisted, which is why I have explained to you really well. Make sure you're not making mistakes on the day of your examination. Now, if apomixis is given, if they are clearly mentioned apomixis, I told you, there can be a case where polyembryony is happening in apomixis. There are conditions where it is not happening, where cells are not coming from the nucleus. Instead, the egg only over here, no, the haploid egg over here, it somehow forms the zygote without the help of the male gamete. Okay, here also, anyway, zygote is being formed. This is, a seed is being formed. A seed is obviously going to be formed since zygote is there. Now, over here, this is an example of apomixis. So, but the egg cell, my dear children, if you know, it's haploid in nature, it's not diploid. The zygote which is there, it is also going to be, sorry, I'm so sorry. Fine. Now, going ahead with the next question out there. Okay, it moved by itself. So, just a second. The secondary oocyte after ovulation is covered by a non-cellular layer of what? So, secondary oocyte which is there, my dear children, you know what? It's so capable that it makes its own layer, but it is not made up of any cells. It is non-cellular, or also you can call it acellular. The structure is known as zona pellucida. This is around the secondary oocyte once it is released. And not just that, apart from the zona pellucida, it also has certain cells, as you can see over here. You call this as corona radiata. But this is not made by secondary oocyte. Only zona pellucida is made by the secondary oocyte out there. So your answer for this question is C. Now, next question. Which of the following is not the function of placenta? Secretes estrogen. Yes, there are 4 hormones being secreted by placenta. HCG, human chorionic gonadotropin, HPL, human placental lactogen, progesterone and estrogen. These 2 hormones are released by ovary also initially, but later on when placenta comes into action, they take up the role. Now, facilitates removal of carbon dioxide and waste material from the embryo. Exactly. It helps in removal of whatever is not needed by the embryo. The fetus. So, it can be carbon dioxide, it can be another waste material out there. Secreted, secretes oxytocin during parturition. Now, oxytocin hormone is not secreted by placenta. I told you there are only 4 hormones secreted by placenta. Oxytocin is released by the female pituitary gland. The mother's pituitary gland, posterior. So, pituitary gland has 2 lobes, posterior and anterior lobe. Over here, the posterior lobe comes into action. PP, parturition. You can remember like that. PP, parturition. Posterior pituitary for parturition. And what does it release? It releases oxytocin. Facilitates supply of oxygen and nutrients to the embryo. Yes, it does. Placenta does the good thing and does the removal of things as well. Okay, provides nutrition and removes whatever is not needed as well. So, your answer for this question would be what? C. Cool. Next, during the embryonic development, which of the following organ is formed first? So, when we die, the first thing that we see is pulse rate. Which is why it means our heart is working or not. That's what we check. Similarly, in the embryonic development stage, my dear children, the first organ to form would be your heart. So, by the end of the first month, you can hear heartbeats out there in the embryo, in the growing fetus out there. So these are the months that you need to remember. First month what happens, second month what happens, third, fifth, sixth and ninth. So 4th is not there. Okay, so you don't have to look around that. So first month heart is formed. Second month you have your limbs and you have your digits. You have your limbs and digits. Third month you get to know what is the sex of the child. That means the external genital organs would be there. The external genital organs would be very much developed, and major organ systems also would be developed. 4th month skip. Then this 5th month the baby says that, God, I need to move around and I need to show my hair off. So basically there will be first movement in the 5th month. First F and 5th also F. So you can remember like that. First movement in the 5th month, and also hair on the head. Hair on the head is also on the 5th month. By 6th month, God says you wanted hair, no, take it on your body also. So there will be hair on the body, as well as the eyelids would be separated. There will be eyelashes formed as well during the 6th month. Now 7th month, 8th month is skipped. 9th month it says that the baby is finally out. Okay, cool. Now going ahead take the snip of this, well, you can get the PDF of it if you join the WhatsApp channel. So if at all you have not joined us, join it right away, my dear children. The link will be given in the description box. Okay. Now, an example of hormone releasing IUD among the following is hormone releasing IUD, intrauterine device that releases hormones. So there are three types of intrauterine devices. One is non-medicated, that does not release anything. It just put inside the body such that our body gets to know something foreign is there, so it will react. So whatever comes along with it, so suppose a sperm also comes, basically it will also be dead because our body is really immunologically very, very active. Okay. Now the second is the copper releasing IUD such as CU7, CUT, all of these things, my dear children, such as multi-load 375. They release copper, and the copper basically does not allow the sperm to move. It just holds the tail of the sperm and says that you cannot move around. Okay, and also it reduces the fertilizing capacity of the sperms. Now LNG20 is what? A hormone releasing IUD. Why it is known as LNG20? Because it releases 20 micrograms of a hormone, of a synthetic progesterone hormone, which you call as LNG, levonorgestrel. So I don't want to get deep into that, but I hope you will remember it better since I have given you certain things. Okay, fine, let's go ahead. Now my dear children, I want it over here. Yes. Next question. Statement 1. Statutory ban on amniocentesis is for sex determination to legally check the increasing menace of female feticide. So statutory ban was put on amniocentesis. Why? Because people used amniocentesis to find the sex of the child, and if it was female, they used to kill them, which is why it was done. So this is correct. This statement is particularly correct. Statement 2. Amniocentesis is a procedure used to test the presence of certain genetic disorders. Is it? Yes, actually it was known for that. Because in amniocentesis, my dear children, what happens? Suppose the baby is here. It is inside a sac which is known as amniotic sac, and over here there is a fluid that is known as amniotic fluid. Now the baby's cells, they shed off, and now you can find the cells in the fluid. Now suppose someone takes the fluid out, they find the cell, they find the nucleus. Inside the nucleus you have the genetic material, and with that you can find the genetic disorders. Be sure you know the genetic disorders. If at all you are asked, can you find jaundice with the help of amniocentesis? Say no, because jaundice is not a genetic disorders. So you should know the name of genetic disorders such as what? Hemophilia, Down syndrome, all of it. Okay, so can I say both the statements are true? Yes. Make sure you're reading the statements carefully. They can twist it and they can say instead of increasing, they can put it decreasing. So make sure you are going through it, and we have faced it during live sessions. So whenever I take live, people get to know where are they doing the mistakes in. Okay, so I request you to join the live that I'm going to take tomorrow, if at all your exam is day after tomorrow, because it is definitely going to help you really, really well. I'm promising you with that. Okay, and my dear children, those of you who are just listening to me and feels that, oh my God, this teacher actually teaches good, then what are you thinking? Click on the like button and share it with your friends as well. Okay, now question number 8. If one parent has a blood group A and the other parent has a blood group B, then which blood group will the offspring have? That means the baby, what blood group will it have? So over here, A and B is there. Listen, A blood group can be like this. It can also be like this. Why? Because A is dominant over O. So anyway, it will be A blood group only. Similarly, B blood group can be like this, or it can be like this, right? Suppose the parent is A like this and B like this. Whatever offsprings will be formed, they will have one A and one B. That means all of them would be AB blood group, right? But you are asked if it is A blood group and B blood group, what are the possibilities? So you have to think wide. It can be a condition like this. And over here you find multiple options. So my dear children, suppose this particular picture, if you look at it carefully, it's IBIO and IAIO. Okay, so this is B blood group, this is A blood group, where O is also there. B is dominant over O and A is also dominant over O. So suppose B came from here and A came from here. B came from here, A came from here, I got AB blood group. Suppose B came from here and O came from there, I got B blood group because B is dominant over that. So AB is there, B is there. Suppose O came from here and A came from here, so IAIO, A blood group will be there because A is dominant over O. I came over here, my dear children, it will be O blood group. So you can see all the blood groups. So this is a wider answer. So please think like that and give the answers. Don't think it will be just like this. No, it can be like this as well. So if at all you have an option like this, go for it. Okay, your answer would be D. Now, going ahead with the next option, Sorry, next question. What is the process of the production of non-parental phenotypes called? What do you mean by non-parental phenotypes? First of all, phenotypes means what? Physical appearance. Non-parental means what? Like we are not looking like the parents. In parents, it's not there. Now there's something new out there. So you can just think of recombination. There is new combinations being formed, which is why there is no sync to the parents. Okay, there is no non-parental phenotypes out there. So your answer for this question becomes what? B. Recombination is the answer. Okay, and since I see all this thing, my dear children, I request you, if at all you have time, go through the 2-marker, 3-marker, 5-marker videos that I have made. It is going to help you really, really well. Okay, if you have time, go through it. Watch it in 2x speed. It will definitely help you. If at all not, then have the PDF, go through it. Okay. Now, going ahead, what would be the distance between the base pairs of DNA whose pitch is 38 Armstrong and roughly has 10 base pairs in each turn? So over here, my dear children, this is known as pitch. So over here, 34 Armstrong is the pitch, and how many base pairs do we have? We have 10 base pairs. So you are being asked, what is the distance between these two base pairs? So if 34 Armstrong is there and 10 base pairs is there, so in Armstrong terms, I can say 3.4 Armstrong. Can I say that? But now over here, do I see an option like that? Sorry, 338 is given, right? Okay, so that is the general size. 34 is the general size, which is why I thought of it. Anyways, 3.8 Armstrong is there, but do I see any option like that? 3.8 nanometer is there, not Armstrong is not there. You have to be sure about the units as well. Now, since it is not there, let me convert into nanometer. And when I convert into nanometer, my dear children, it comes out to be 0.38 nanometers. Okay, it comes out to be 0.38 nanometers. So 1 Armstrong is equals to 0.1 nanometer, my dear children, which is why we can just have to, do the calculation, and it comes to be 38 nanometer. So this is your answer. Cool. Okay, so Armstrong is bigger, nanometer, you can think like that. Okay, let's go. Next. The role of RNA polymerase 2 is transcription of RNA polymerase, it does transcription of DNA to RNA. It forms RNA, right? Now, what type of RNA is also there? Because we have multiple RNAs. rRNA is ribosomal RNA, the RNA which is found in ribosomes. mRNA is messenger RNA that carries the messages and go out to the cytoplasm. tRNA is transfer RNA, basically, which helps in the process of translation. And hnRNA is heterogeneous nuclear RNA. So, when we say transcription has happened, the first thing that we get is hnRNA. You know what? It's immature RNA. Why do you call this immature? Because over here, whatever was there in the DNA is transcribed totally over here. So there might be regions that codes for proteins, that is known as exons, and there are regions that do not code for any proteins, they are known as introns. So actually they are of no use. Introns are of no use. We have to remove it. So we make this hnRNA undergo lots of procedure to make it convert into what? Mature RNA, which is known as mRNA or the messenger RNA. There is splicing that happens where you cut the introns. There is capping, there is tailing, all these procedures happening, and after that you get the mRNA. Now the question is, what is role of RNA polymerase 2? The role of RNA polymerase 2 is this. It helps in transcription of this particular hnRNA. It helps in formation of hnRNA. So you have 3 RNA polymerases. The first one is, it is going to transcribe the rRNA. So you can remember like this, RNA polymerase. The priority will be to look for rRNA because everything is RR. So rRNA is the first thing that is going to be formed. RNA polymerase 2, it looks for next priority, which is what? Formation of messenger RNA. Now the third priority is what? Third priority, TR. T for third and T for tRNA. So that is the third priority. And apart from it, certain types, please remember, certain types of rRNA are found over here exclusively. That is 5S rRNA and different type of RNA, that is snRNAs. Okay, 5S rRNA. Over here we have all the rRNA being formed, but over here exclusively 5S rRNA, and then snRNAs are also being formed by the RNA polymerase 3. So this is very important, go through it. Now going ahead, Miller in his experiment synthesized simple amino acid from. So how did he get simple amino acid from? By doing a simple experiment which is known as Miller's experiment, right? What all did he use? As you can see over here in this particular picture, this is CH4 is what? CH4 is methane, and then NH3 is what? Ammonia. H2 is what? Water. H2 is what? Water vapor. Okay, you can remember like that, or basically not water vapor, hydrogen. So over here H2 is vaporized form of water, and my dear children, hydrogen is also being used, that is H2. So these are the few things that you need to remember. I'll show you the picture for better clarity. 4 things, okay, in the Miller's experiment, and that was this. So your answer would be hydrogen, methane, ammonia and water. Hydrogen, methane, ammonia and water. Cool. Let's go ahead with the next question. In some animals, the same structures develop along different direction due to adaptation to different needs. This is called. So my dear children, what is happening? Because of different needs. Okay, there are multiple needs by the organism, which is why they are the same structures which are there, they are evolving into different things. The best example is our forelimb. So we have forelimb like this. The cheetah is using it for what? Running. Okay, the whale which is there, it is using for swimming. The bat which is there, it is using for flying. So because they are in different habitats, the same structure, our forelimb, the same type of bones and everything out there, my dear children, has been used differently. This is known as divergent evolution. You are diverging. You're not coming to a point, rather you are diverging. You're moving away because of different habitats, different adaptation. So this is known as divergent evolution. Convergent evolution basically means you are not related. You don't have any common structure, but still you are behaving to be the same thing. For example, birds and butterfly. Bird is coming under aves, and this butterfly is an invertebrate. Okay, it's not even a vertebrate, it's an invertebrate. So over here, butterfly has wings to fly. Similarly, birds also have what do you call the wings to fly out there. So that is convergent evolution. Since both of them are living in the same environment, that is the what do you call in the air. So over here, it's divergent evolution. Because of different needs, they are diverging. Cool. And all this thing not needed. Going ahead, amoebic dysentery is caused by amoebic dysentery. Dysentery means whenever then in the stool there is in the stool there is mucus as well as blood. This is known as dysentery. Now an amoeba is causing it. What is the name of this amoeba? Antamoeba histolytica. So E. histolytica would be an answer for this particular question, that is amoebic dysentery. Okay, it's not E. coli, nothing else. Now, which of the following is not a lymphoid tissue? Not a lymphoid tissue. Spleen, tonsils, thymus, pancreas. Pancreas is not. Spleen is a secondary lymphoid tissue. Tonsil is also secondary lymphoid tissue, and thymus is a primary lymphoid tissue. What do you mean by primary? This is where the cells are basically going to mature. Mature into what? Antigen sensitive lymphocytes. They are going to be sensitive. They don't act there. They don't do dushum over here. Where do they go to do dushum, my dear children? They go to spleen and tonsils, where they become effector cells. So these terms are really important. Effector cells. Where? Here. What happens over here? They become antigen sensitive. They know that I have to be sensitive to this particular antigen and I have to kill it later when I go to the secondary lymphoid organs. That's all. And one more primary lymphoid organ is there, which is known as bone marrow. That is where the cells are being formed as well as matured. Okay. So in thymus, T lymphocytes go and mature. In bone marrow, everything is being formed. The B lymphocytes, the T lymphocyte, everything is being formed. B lymphocyte mature there only. T lymphocyte come here for maturation. Okay, now next thing, fill in the blanks by choosing the appropriate words from the given thing. The female gametophyte of an angiosperm is known as, what do you call the female gametophyte, my dear children? Gametophyte means the one that will be having the gamete within it. It is going to give rise to the gametes. The female gametophyte is known as embryo sac. Okay, now dash is implanted in the uterus. What is implanted inside the uterus, my dear children? It would be a stage of embryo. What do you call the stage? You call the stage as blastocyst stage. What is so special about this stage? So there are cells like this outside, and there will be a mass of cells over here inside. This is known as inner cell mass, and this is known as trophoblast. And there is a blastocele. There is a cavity over here which you call as blastocele. Together, all of it you call this as blastocyst. So this particular thing goes and get implanted into the endometrium of the uterus, my dear children. And you know what? Trophoblast is going to help in the formation of placenta as well. Whereas the inner cell mass strictly focuses on what do you call making the embryo grow bigger. Okay. So over here, blastocyst is implanted. Blastocyst. cyst is implanted. Now sterilization procedure in human females is, sterilization basically means it's a what you call surgical procedure where you're going to cut the fallopian tube of the female such that the ovum is not transported, such that the the sperm never meets the ovum. I know it's cruel, but that happens. And what do you call this procedure as? It is known as tubectomy, since you're cutting the fallopian tube. So rather than writing that over here, I'll just write 18. For 18, the answer is this. Okay. Now, dash is an example of point mutation. That means only at one particular point, a single base pair is being changed. A single base pair is being changed. For example, suppose it was GAG, but now it is GUG. Do you think it will bring out certain changes? Yes. GAG codes for a protein that is, amino acid that is known as glutamic acid, and over here, this guy codes for another amino acid that is known as valine. This is an example of sickle cell anemia. A comes before U, so always remember A was there and it was replaced by U. G comes before V, so G was there, glutamic acid was there, and it is replaced by valine. And it is going to hamper the structure of beta hemoglobin chain. Okay, so hemoglobin is made up of alpha and beta globin chains, so it is going to hamper the structure of beta hemoglobin chain. Fine. So this is an example of point mutation. So where is it written? Sickle cell anemia is the answer for this question. 19th. Next, additional stability to DNA when compared to RNA is due to the presence of. Why do you think DNA is more stable than RNA, my dear children? Because it has uracil? No. RNA has uracil. DNA has what? Thymine. What's difference over here? What's the difference over here? Any idea? There is a methyl group that is being added, my dear children. There's a methyl group that is being added to the uracil to make it thymine, which is why thymine is also known as 5-methyl uracil. So your answer for this question is thymine, not uracil. You're asked about why DNA is more stable. Okay? Next questions are there. So now we have the 2-marker questions. So listen to me carefully. We will be going fast only because I know your time is so precious as of now. So write 2 events that occur in the ovary and the uterus during the follicular phase of the menstrual cycle. So the first phase is menstrual phase, where the endometrium which is there, it falls off. It goes off, right? It degenerates. That's the term that you use. And after that, what happens? Basically, the next thing happens, which is known as follicle development. The follicles which are there, they are going to develop. Under the influence of what? FSH and LH hormone, follicle stimulating hormone and luteinizing hormone. Now when the follicles are developing, think around it, okay? Just imagine, follicles are developing, you know, the cells of the follicles, they are going to release a hormone which is known as estrogen. And what does this estrogen do? Estrogen slowly, whatever endometrium just fell down, slowly regenerates it. This estrogen regenerates, which is why you call this as proliferative phase also, because the endometrium is slowly getting proliferated. So in the ovary, what is happening? The primary follicles which are there, as I told, it is maturing into Graafian follicle, which is the mature form of follicles out there. It won't form lot of Graafian follicle, only one. Okay, so multiple primary follicles are going to get matured, but it will be said, hey, you stop. Only one can become what? Graafian follicle. So this is happening during the follicular phase within the ovary, and not just that, as I told you, the growing follicle secretes estrogen, which helps in what? Proliferation of the uterine lining. So what happens in the uterus? The endometrium regenerates and thickens after menstruation, which is why it is known as proliferative phase as well. Cool. So that is the 21st answer. Now, going ahead to the next question, mention any 4 sexually transmitted diseases. Okay, 3 of them very easy, starts with H only. Hepatitis B, genital herpes, and human immunodeficiency virus, that is HIV. So HHH. Apart from that, syphilis you can remember, or if you want gonorrhea you can remember, chlamydiasis you can remember. All of them are what? Sexually transmitted diseases. Now you would be asked this question as well, that is, what are those 3 diseases which are not completely curable whatever you do? So as I said, all of them start with H. Hepatitis B, genital herpes, and HIV/AIDS. So this is what you need to remember if the question is framed like that. Going ahead with the next question, how do the following assisted reproductive technologies assist the couple to have children? ZIFT and GIFT. What do you mean by ZIFT? Zygote intrafallopian transfer. What do you mean by that? Zygote is being put. Intrafallopian means inside the fallopian tube. So ma'am, only zygote is being put. Where do you get the zygote from? The zygote is made in the laboratory condition. This is an example of IVF, in vitro fertilization. In vitro means in the lab fertilization is happening. So egg is taken, the sperm is taken, it is fertilized, you get the zygote. Now either you can put the zygote in the fallopian tube, or also you can put the embryo, but there is a catch. If at all you want to put it in the fallopian tube, the embryo just has to be up to 8 blastomeres, not beyond beyond that. If at all it is beyond 8 blastomeres and you put it there, what will happen? The whole baby will be formed out there. This is known as ectopic pregnancy. This can lead to very big problems in the case of the mother as well as the child. So up to 8 blastomeres, embryo up to 8 blastomeres, or the zygote, my dear children. Zygote is single cell. The moment it divides, it forms the embryo. So up to 8 blastomeres, it can be put into what? Into the fallopian tube. This is known as zygote intrafallopian transfer. Fine. Now, it is basically done where normally fertilization is not happening within the female body, but they have all the suitable conditions for growing the baby out there. Now, gift is there. What is gift? Where a female is gifting another female a gamete. Maybe because that female is not able to produce a gamete by herself. So, she is not able to produce her own ova, my dear children, or there might be some problems with the ova, which is why the gamete is transferred inside the fallopian tube. This is known as gamete intrafallopian transfer. Ova collected from a donor are transferred into the fallopian tube of another female who cannot produce eggs but can provide a suitable environment for what? Fertilization and further development. That is known as gift. Okay, so if at all you are asked about gift, write this. If at all you are asked about gift, write this. And always remember, if it is more than 8 blastomeres, you put it inside the uterus, and that is known as intrauterine transfer. That is known as intrauterine transfer if it is more than 8 blastomeres. Cool, done. Fine. Let's go ahead. What is MTP? Mention the safe period for MTP. MTP stands for medical termination of pregnancy. Okay, that means you're terminating the pregnancy. Now, it is intentional or voluntary termination. Okay, it's not like I do not know abortion happened. No. Abortion can happen naturally or it can be induced. So, this is induced abortion. It is a voluntary thing. You know it. You are doing it intentionally. And why? Probably because the baby might have some genetic disorders, or it might be fatal for the mother as well as child to have the baby, or it might be a result of unprotected coitus, my dear children, especially during rapes. Okay, so you might be asked when it is, when do you think it's right to do MTP? That question can also be framed. Okay, or yeah, like that. So now the next part of the story is, what is the safe period of MTP? Now this can be confusing. Listen. It is considered to be safe up to 12 weeks. That means 12 weeks means what? 3 months. 12 weeks means 3 months. 3 into each month has 4 weeks, my dear children, which is why 12 weeks. So until 3 months, it is considered safe. However, it can be performed until what? 24 weeks as well. Up to 24, that means 6 months. 24 weeks. So it can be performed, but it is considered to be safer during until 12 weeks, but it can be performed. Have that in your head. Can be performed up to 24 weeks. So if you're doing it 12 weeks, then you need just one medical practitioner's advice. If it is a 24 weeks, since it is very much risky, you need 2 medical practitioners advice. Now over here, the things that you need to remember is the year when was it launched, the Medical Termination Act was there, so many years are being mentioned. Make sure you're looking through it, very, very important. Okay? Right? Any 4 symptoms of Down syndrome. Down syndrome is caused when what happens, my dear children? There's an extra chromosome. Which chromosome? 21st chromosome. So this is known as trisomy of 21st chromosome. Since you have 2 chromosomes, 1 chromosome comes extra. That is why trisomy of 21st chromosome. You would have short stature. There will be small round head. The furrowed tongue will be there. Tongue will be like a kind of little bit furrowed, and partially opened mouth will be there. Broad palm will be there with characteristic palm crease. It can be the lines which are there clearly visible. Okay, and physical, mental and psychomotor retardation would also be there. So short stature, small and round head, furrowed tongue, partially open mouth, and all these features are also something that you need to remember. Okay, and it's trisomy of 21st chromosome. Regarding all the chromosomal disorders, you should know certain terms. Aneuploidy. What is aneuploidy? You should know what is monosomy. Monosomy means you have lost 1 chromosome. Trisomy means you have gained 1. Right? Monosomy's example is Turner syndrome. Trisomy's example is this, as well as when it happens in the X chromosome, you call that Klinefelter syndrome. So all of these symptoms are very important, and their karyotype. That means how many number of chromosomes are there. All of these things. So as I said, I have put it in several videos. Make sure you go through it, because if at all I teach over here, this video is going to be pretty long and we would be deviating from the question paper as well. Okay. Now what is adaptive radiation? Give an example. Adaptive radiation basically means, just imagine, okay, I am a finch, a bird. this finch which was there, I had some, in my group, some finches has some mutation. By themselves, they had some genetic mutations because of so environmental reasons or things like that, which is why they were kind of different. Now, when these different finches, they went to that particular site, they were able to find that I am able to eat insects better rather than what I was eating before, because my beak and everything is changed because of the mutation. So now they have moved to that particular site and they have located there. Similarly, some other group of finches went there, some other group of finches went there. So they have radiated to all sites, and when they moved there, they got to understand that I am better adapted to this particular place. This is known as adaptive radiation. I can give an example of it. Look over here. The finch I was talking about. So there are tree finches, there are warble finches, there are ground finches. So they have different type of beaks, focusing on different things. When they moved away because of the mutations, they moved away, and they found out because of the mutation they had different beak size, different beak shape, and which is why I am better adapted over here. So this is an example of adaptive radiation, and it was found by Charles Darwin. Okay, so it is a process of evolution of different species in a given geographical area. That is very important. This is restricted to a given geographical area. Where was all this happening? In Galapagos Island. Okay, so yeah, starting from a common ancestor. So they will have a common ancestor and showing adaptations to different ecological niches. So there will be different ecological niches and they show better adaptation to those. Example, Darwin finches on Galapagos Island. They evolved from a common ancestor but developed what? Different types of beaks to occupy different feeding habits. Okay, so this is one very important concept out there. Now what are the, what type of evolution is this? What type of evolution is this? This is divergent evolution, because they are diverging to different area. Okay, suppose now over here you had birds, you had something else also. You had something else, and they are also diverging like this. So they are also radiating like this, both of them together. So now what will it be? An example of convergent or divergent evolution? I am telling, there is a species. Okay, they have moved to different areas. That is an example of divergent evolution. Agreed. There is a B species of organism, not finches, something else. They are also moving like this, and they are also getting adapted. They also have certain features like the A1. So what example is of this? What is this an example of? That is the question, sorry. It is an example of convergent evolution. So two different species converging to a same point and what we call feeding on or having the same type of habits. That is an example of convergent evolution. Individually, if we talk, they are showing divergent evolution. But if we talk about two species, they are showing what? Convergent evolution. This is an example of placental mammals and marsupials. Go through it, very important. Now, what are the applications of DNA fingerprinting? What do you mean by DNA fingerprinting? Our DNA is very unique. Suppose I did a crime, okay, and there is some blood or some sample of my hair follicle or things like that, my dear children, and you get the entire coding of the DNA being done, it will be found that I am the person, because my DNA is unique. You do not have my kind of DNA. This is known as DNA fingerprinting. So what is the use of DNA fingerprinting? First thing, forensic identification helps in identifying criminals in rape, murder or theft cases. Second, paternity or maternity disputes, because we do not know who is the actual parent of this particular child. Now you do this test, you do the DNA fingerprinting and you get to analyze that. So we would be having certain things common in between us and our parents in the DNA. So with that we will be able to find it out. Now identification of missing individuals. Again, if I have the DNA sample, I know this was a thing, and from some missing individual I find the same type of thing, I get to understand. Now genetic diversity and evolutionary studies used to study variations among individuals and population. So you get to understand that how varied are we, how we got different and things like that, because we would be having some common things and some different things as well. Okay, so now the process of DNA fingerprinting can also be asked. To make sure you're going through that. Again, as I said, it's in the video. Watch it. Now, answer any 5. Okay, now we have entered the 3 marker question zone, my dear children. We have to be very attentive over here while answering the questions, and also briefly explain. The first question is briefly explain the structure of male gametophyte. What is a male gametophyte? It's the pollen grain, my dear children. And in pollen grains, what is the thing that you need to remember? First of all, write it down, whatever things are there. Okay, and second, explain with the help of a diagram. So this is a pollen grain, or you call as male gametophyte. It has 2 types of cells. One is the vegetative cell, this is the vegetative cell, and this is the generative cell. The vegetative cell, my dear children, it has irregular nucleus, large irregular nucleus, and this guy has an elongated spindle-shaped nucleus. Now, this is a question. What is the shape of the nucleus? So here it is irregular, here it is elongated. Now, very important thing, in the cytoplasm of the vegetative cell lies the generative cell. So, in the cytoplasm of this big cell lies the small cell that is known as generative cell. Remember that as well. You can get a question that generative cell has bigger, cytoplasm, and in that comes vegetative cell. That's a wrong statement. It's ulta, right? Now, the generative cell which is there, it will generate what? It will generate gametes. How many? 2. It forms 2 gametes. So again it will undergo mitotic division and will have 2 cells out there, 2 gametes out there. Now it has its own plasma membrane. Apart from that, there is one layer which is known as intine. Now intine which is there, it is made up of cellulose as well as pectin. Cellulose as well as pectin. Priyanka Chopra, you can remember like that. PC or CP, whatever way you want to remember, you can remember like that. Okay, cellulose and pectin. It is continuous layer. There is no anything, nothing, no gaps out there. And then you have a discontinuous layer out over here, which is known as exine. An exine is made up of one of the most important material out there, which is known as sporopollenin. Okay, sporopollenin. It is the toughest material. If you put acid, alkali, anything, it won't get destroyed. So that is the thing for exine. Now, there are some pores over here. Wherever sporopollen is not there, there is a pore. You call this as germ pore. And this is where the pollen tube arises from. So the pollen tube is made from the vegetative cell itself. The vegetative cell which is there, it forms the pollen tube, and in that you will be having the 2 gametes out there. So, yes. So the male gametophyte in flowering plants is pollen grain, and it has 2 cell structure consisting of vegetative cell, that is also known as tube cell because it helps in pollen tube formation later on, and generative cell which forms the gametes. Now you talk about them. Vegetative cell is larger, has abundant cytoplasm, and has a nucleus. What type of nucleus? Irregular nucleus. The generative cell is smaller, floats in the cytoplasm of the vegetative cell, and divides to form 2 male gametes during pollen tube growth. Okay, it forms 2 male gametes. Now, my dear children, 60% of the plants are angiosperms, okay? The flowering plants are going to release the pollen grain in 2 cell stage. That means there will be vegetative cell and there will be generative cell. The generative cell has not divided yet. In 2 cell stage, majority of them in 2 cell stage. 40% of them which are remaining, they shed their pollen grains in 3 cell stage. That means the generative cell has divided into 2. So 1, 2, 3 cells will be there. Okay, so these are the things that you have to write. Make sure you make the diagram properly and explain it. That's all. Now, clistogamous flowers are invariably autogamous. Justify the statement. First of all, what do you mean by clistogamous? Clistogamous means closed flowers. They are closed flowers. They have the female part and the male part, but they are closed. Now, autogamous. Autogamous means over here, the pollen grain of the same flower lands on the stigma of the same flower. This is known as autogamy. Autogamous nature, okay, and this is also known as self-pollination. So now you are saying the closed flowers, whatever you do, they are going to be autogamous only. There is no other option. Is that true or false? Yes, it is true, because they are closed. Do you think any other flower pollen grain can come and land over here? No, Baba, they're closed. Now you have to justify the statement. So you have to say that it remains closed, and the stigma and the anther are enclosed within the same flower. Pollen grains from the anther directly comes in contact with the stigma of the same flower. There is no chance of cross pollination because it is closed. Hence, clistogamous flowers are invariably autogamous. That means self-pollinated. Example: Viola oxalis commelina. Viola oxalis commelina. You can explain about certain extra things, such as over here you don't have to be worried about pollination anyhow. Seed setting will be there. Seed will be definitely formed. There is no need for pollination. But what is the drawback of it? Drawback is that, my dear children, pollen grains of the same plant, stigma of the same plant, everything of the same plant, which is why there is no genetic variations. Genetic variations will be less. So you can explain like that. Okay, so Viola oxalis and commelina, they don't just have closed flowers, they also have open flowers, and the open flowers are mentioned over here. They are open, they do cross pollination and everything out there. Okay, they can show self pollination, they can show gytonogamy. That means within the same plant, suppose 2 flowers are there, that is cross pollination, but not true cross pollination. So gytonogamy can happen, and also xenogamy can happen. They can pollinate with other plant as well. So clistogamous flowers are towards the root or the rhizome region, whereas chasmogamous flowers are towards the shoot region. Okay? Cool. Next question. Describe any 3 outbreeding devices that prevent autogamy. Okay, so we do not want self-pollination to happen. What can be done? There are 3 different ways where plants avoid it, because why? It causes inbreeding depression. That means the same type of gametes, whatever toxic traits were there in the parent is there in the progeny also, then the progeny dies because it does not have any good trait. That is known as inbreeding depression. Now to avoid that, the plant have outbreeding devices. So the first time, different time of maturation. Suppose over here the female anther is maturing late and the stigma is maturing early, then obviously it can prevent self-pollination to an extent. So that is one thing. It is also known as dichogamy. Second is positioning of stigma and anther. Suppose over here, the stigma is at the top, okay, over here, but the anther is here. So now, do you think pollination can happen easily? No. So positioning of stigma and anther. The third thing is self-incompatibility. Now this is a genetic mechanism. That means, suppose the pollen grain landed over here of the same plant. The plant itself does not allow, there would be something happening over here in this particular region such that it does not allow the formation of pollen tube growth. This is known as self-incompatibility. Okay, a genetic mechanism that prevents the pollen grain from the same flower or the plant from fertilizing its own ovule, thus promoting what? Cross-pollination. Okay, it won't allow that to happen. So these are the 3 things. Apart from that, there is also production of unisexual flowers. Unisexual flowers. That means in a flower there won't be 2 sexes, only one sex. Suppose it is a female flower and this can be a male flower. If at all both of them are on the same plant, then you are doing the same thing. Then again it can come and land over here. This is known as monoecious plant. This is known as monoecious plants such as castor, coconut and things like that. But still it helps in some ways. Apart from, if at all they are in the same same flowers, both the male and female is there, then there are more chances. Here the chances are at least little less. So this is monoecious plant with unisexual flowers. There can be monoecious plants with both the bisexual flowers as well, where both the things are there. So this is an example, and apart from that, major example is dioecious plants. Dioecious means, my dear children, there's a plant with what? A female flower. Only female flowers will be there in this plant, and there will be another plant with only male flowers. So sexes are completely separate. That is example of dioecious plant. And the major thing that you need to remember is papaya comes under this. So unisexual flower formation is also one thing. In that you can have dioecious, example is papaya, or it can be monoecious, but in monoecious it can still happen, but it can limit to certain limits. Certain levels, that's all. Cool. Chalo. Next, write the schematic representation of spermatogenesis. Are you writing? So how do you write it? Schematic representation of spermatogenesis. First of all, you have spermatogonia, my dear children. Okay, you have the spermatogonia. The spermatogonia, my dear children, it undergoes a process of differentiation. That means it feels like I have to mature into something that can form sperms. So it matures into primary spermatocyte. So until here it is diploid only, 2n number of 46 number of chromosomes are there. Now it undergoes a process of meiosis. And in meiosis, my dear children, first meiosis itself, my dear, the number of chromosomes become reduced to half. So the thing that you get from primary spermatocyte, you get secondary spermatocyte, and over here they are haploid. Once meiosis has started off, the results that you will get, they would be haploid cells. Now again these haploid cells are undergoing meiosis too, because meiosis happens in 2 stages. Again, do you think reduction will happen? No. It will be exactly the same. Over here the things that you get is known as spermatids. You know what? They are like kids. They are immature. Small kids, no, they are immature. They are round in shape, but the sperm shape is what? Elongated. That's how it can undergo fertilization. Otherwise it won't be able to move, right? So this spermatid has to convert into spermatozoa. That is, this forms, my dear children, spermatid, kid, okay, into spermatozoa. What do you call this process as? Write it down. This process is known as spermiogenesis. Spermiogenesis. What is the entire process known as? Spermatogenesis. This is spermiogenesis. And at the end, when the sperms are completely formed, they will move out of the seminiferous tubules where they were formed. This process is known as spermiation. Aviation, you can remember like that. Spermiation. Okay? Cool? Done? Chalo. Explain the neuroendocrine mechanism of parturition. That means the signals of parturition, the movement of baby outside the the womb, comes from neuronal signal as well as endocrine. Endocrine means what? Endocrine means hormones. Hormones are also playing an important role and neurons are also playing an important role. How can you prove that up? So my dear children, first of all, you need to understand the first signals come from both the fetus as well as placenta. Remember this very important question. This is known as fetal ejection reflex. What do you call this? Fetal ejection reflex. This comes from both fetus as well as the placenta. So both of them combined together are going to release some hormones. They are going to release some hormones. Assume it's prostaglandin. You don't need to remember the name of the hormone, I'm just telling you. Assume there's a hormone, prostaglandin. You know what this prostaglandin does? It causes mild uterine contraction. So mildly it causes uterine contraction, and when it does, the baby will try to move out. And when the baby tries to move out, you know what? In the cervix region of the uterus, my dear children, there are certain receptors. Those receptors immediately tells the brain. So there is a neuronal signal going up now. So hormones are released, then now the stress receptors are active. It sends a neuronal signal to where? To the maternal pituitary. So the mother's pituitary gland, I told you PP, remember? Maternal pituitary gland, posterior pituitary, it releases a hormone which is known as oxytocin. Now the hormone, now we have hormone. Until now it was neuronal. First it was hormonal, then neuronal, then again hormonal. The oxytocin will now come where? In the uterus. Because in the uterus there are so many receptors lined up for oxytocin. So suppose in the uterus there is a layer that is known as myometrium, the muscular layer. It is made up of smooth muscles. This myometrium has certain receptors which you call as oxytocin receptors. They actually wait for oxytocin. So when the oxytocin comes over here, it will cause what? More uterine contraction, and finally the baby would be moving out. So that is the thing. Fetal signals from the fully developed fetus and the placenta initiates what? Mild uterine contraction. You don't have to mention prostaglandins. Now these contractions sense what? Neuron. You don't say that receptors are active, this and that. You directly say that this signals what? This gives neuronal signals to the maternal pituitary, stimulating the release of oxytocin. Now the oxytocin comes and strengthens the uterine contraction, which further increases what? Neural signal. Again the baby tries to move out, again the signal is active, and this goes on, and this is known as a positive feedback loop. Positively it is happening again and again and again until the baby and the placenta moves out. Okay, so will you be able to explain it? I hope you do now. I hope you have that imagination happening now in your head. My dear children, are you liking this video? Do you think it is helpful in some ways to you? If at all you think, as I said, subscribe to the channel right now and also click on the like button. Okay, and let me know if it is helpful for you, if it's not, whatever is there. And if you have doubts, definitely ask me out. Okay. Now, what is immunity? Mention the 4 barriers of innate immunity. What do you mean by immunity? Immunity is a process that protects us from what you call any diseases. Any diseases out there, it is going to act like a barrier. Now, what is innate immunity? Innate immunity is a specific type of defense mechanism that we have, which is, it is a non-specific type of defense. What do you mean by non-specific? Basically, for example, I'll give you basically, for example, you have skin. Now, skin won't say that, hey, virus, you don't come in, bacteria, you can come in. Can you do that? No, skin is common for all. Whatever is coming over here, is it specifically saying anything to anyone? No. Is it specifically rejecting something and accepting something else? No. So it is non-specific. It is going to be common for all. For example, the mucus which is being secreted in our body, do you think it just traps small organisms or can trap anything out there? It will definitely trap what? Anything out there, which is why it is known as non-specific type of defense. It is present from the birth. You don't get it, okay, throughout your lifetime. That is known as acquired immunity, opposite of innate immunity. This is present from your birth, from the day you are born. Okay, and it will help you to protect against all the pathogens. So it is a non-specific type of defense present from birth that provides the first line of protection against all type of pathogens. The 4 barriers of innate immunity that is being asked. The first one is physical barrier, your skin and your mucous membranes. It prevents the entry of microbes, which is why it is a physical barrier. Physiological barrier, that means the secretions out there. For example, saliva. Saliva has an enzyme, lysozyme. Okay, please write it somewhere, lysozyme. Saliva, tears, everything has an enzyme that kills the bacteria or breaks down the wall of the bacteria, which is made up of peptidoglycan. And apart from that, you have another secretion in your stomach, HCl, hydrochloric acid, my dear children, that destroys other microbes as well. Now you have cellular barriers, such as what? White blood cells. Okay, white blood cells which are there from your birth, not all type of them, at least some. For example, neutrophils. So neutrophils are also known as polymorphonuclear lymphocytes or leukocytes. Do not forget it. Why they are known like that? Because, my dear children, over here they have their nucleus in different shape. It is morphologically very different and it has multiple lobes out there, which is what is known as polymorphonuclear leukocytes. So neutrophils, macrophages, all of them have a common function, and that is to do phagocytosis. What do you exactly mean by phagocytosis? Phagocytosis means basically taking in something which is very toxic or which can cause problem. It takes it in and breaks it down. That is known as phagocytosis. So neutrophils is there, which is also known as polymorphonucleocytes, and you have macrophages which does phagocytosis. They act like cellular barriers. Apart from that, you have natural killer cells as well. Okay, natural killer cells are there targeting other cells getting infected by viruses and things like that. So all of them have different functions, but you need to remember it. So monocytes are the cells which later develop into or which give rise to macrophages later on. So you need to remember all of the cells. So they are what? Cellular barriers. Apart from that, you have something else which is known as cytokine barriers. What do you mean by that? So certain cells which are infected by something, they would be releasing something else which are known as interferons, or they will be releasing certain things that can save other cells from being infected. For example, this cell is being infected by a virus. Now this cell is releasing a cytokine, that is interferon. Okay, and what does the interferon do? Tells the other cell to be safe. If there is a virus attack in our body, you have to be safe. So those are an example of cytokine barriers. So virus infected cells secretes interferons which protect nearby healthy cells from further viral infection. I hope that is clear. Okay, cool. Next, name the 6 stages of human evolution in correct sequence. So this is evolution chapter related question. Over here, the first thing that you need to remember is Dryopithecus and Ramapithecus. So there were 2 type of organisms, Dryopithecus, Ramapithecus. Ramapithecus was more man-like, whereas Dryopithecus, if you see, my dear children, it was more ape-like. Okay, Ramapithecus, Ram, you can remember like that, Rama. Okay, Ramapithecus more man-like and Dryopithecus more ape-like. Okay, and over here they were found 15 million years ago. After that you have Australopithecus. Australopithecus, my dear children, they walked upright. So until now they were like not walking upright. They used all their 4 limbs out there for walking. But from here onwards they were walking on 2 legs. Not completely erect, but somewhat like that. Okay, and the height around 4 feet. They used stone stools and ate fruits. They didn't eat meat and all, they ate fruits. Now come Homo habilis, the first Habibi in the town. Okay, 2 million years ago, the first human-like hominid, the first human-like feature, Homo habilis, came 2 million years ago, and their brain capacity was 650 to 800 cc. Cranial capacity was 650 to 800 cc. Again, as I said, they didn't touch meat. It was only fruits. They relied on fruits, vegetables. Okay, and they used tools. They used to form the tools, my dear children, stone tools. Now, after that, they finally became what? Erect. The human beings, they stood erect. So that was Homo erectus 1.5 million years ago. They had larger brain. So from 650 to 800, it has become 900 cc. And they started eating meat as well. And they started using fire also. And from them, from Homo erectus, came 2 other things. That is Neanderthal man and also Homo sapiens. So Neanderthal man was the first thing to come up there. So Neanderthal man were found almost 1 lakh to 40,000 years ago, and their cranial capacity was the highest. If you see, the cranial capacity is increasing, right? It was now 1400 cc. And the best part of the story that you need to remember is Neanderthal man were the people who first time used the clothes to hide their body and also started burying their dead. That means there's an emotional thing out there. They started burying their dead and also used hides for their body. Now, as I said, from this came 2 of them. This came a little later, that is Homo sapiens, the modern human beings out there, almost 75 to 10,000 years ago, my dear children, originated in Africa and then later spread worldwide. Okay, so that is the thing that you need to remember. If at all you forget the flow, remember like this: daring apes have evolved new skills. So D for Dryopithecus, but you have to remember humans came from Ramapithecus. And A for Australopithecus, H for Habibi, that is Homo habilis, and E for Homo erectus, and N for Neanderthal man, and S for sapiens, Homo sapiens. Okay, works. Going ahead, but before I go ahead, very important thing, Have you joined to our crash course? So we have launched the KCET 90 crash course. This is a 90-day crash course which is targeted for 2 exams, that is KCET as well as COMEDK. And we are going to be here for you to solve everything out there, whatever queries that you have. So if at all you wish to participate in this, definitely call on to this number or look at the link given in the description box. You know what? Initially we thought we will make it offline, but seeing your request, we have made it online as well. So those of you who feel comfortable to come to Deeksha campuses and study, you can take the offline version. If at all you're comfortable learning online, you can take the online version as well. Okay? Chalo. Now going ahead with the next question out there. Now these are some 5 marker questions. Okay, and very, very important out there. We have to take 4 of them and do. So draw a neatly labeled diagram of the sectional view of the male reproductive system. So actually sectional view is this. But in the exam, they focus on this particular thing. I know it's wrong to say that, so I'll definitely recommend you to ask to your teacher, whoever is going to check your paper, to ask them once, what if this question comes, what I have to draw? So actually it's this, but usually what is being made is this, so definitely go through it, look out for all the labelings as well. Okay? Next, state the law of independent assortment illustrated with the schematic representation in a pea plant. So what do you mean by independent assortment? Independent assortment says that if you have, just give me a second, okay? Okay, so we were here, we had to talk about the law of independent assortment, my dear children. And I'll just explain you with the help of a quick little diagram, okay? So over here, you have round and yellow plants, and you are fusing it with what? You're fusing it with, or basically you're crossing it with what? Wrinkled and green plants, right? So over here, round and wrinkled is talking about what? The shape of the seed. Yellow and green is what? The color of the seed. So there are 2 different traits. One is the shape as well as other one is the color. And now over here, if I say round and yellow, I'm taking round and yellow and wrinkled and green, and I'm crossing it up. What do I exactly mean over here? So I have 2 chromosomes, capital R, capital R, capital Y, capital Y. That is what I mean by capital R, capital R, capital Y, capital Y. Similarly over here also, it would be small r and small y. Cool? Now my dear children, look at this particular thing very carefully. Whatever gametes you are forming, from this pair, one can come in. From this pair of characters, one gamete, one allele can come in. So anyway, it is capital R. So whatever you do, you will get just capital R. Similarly over here, anyway, whatever you do, you will get capital Y only. So one chromosome having capital R, another chromosome having capital Y. Similarly over here, you will have one chromosome containing small r, another one with small y. Cool. Now whatever gametes which are being found, if at all you try to fuse it, it will be what? It will be capital R small r, capital Y small y. That means now the scenario is what? You have got capital R from here and you have got another chromosome with small r from here. Similarly, you have got capital Y from here and you have got small y. So this is a new set of chromosomes that you have got in the progeny. All of them would be what? They are round and yellow because capital R is dominant over small r and capital Y is dominant over small y. So they have to be what? They have to be round and yellow. Right? Now, this is first generation. I am going to experiment with the second generation to find out the real thing. So for that, again, what will I do? Let's see. So capital R small r, capital Y small y, and capital R small r, capital Y small y. This is the scenario as of now. So the chromosomes are lined up like this. Okay, from here one can come, it can be capital R. From here one can come, it can be capital Y. Again, as I said, from here capital R can come and here small y can come. There are chances. From here small r can come, from here capital Y can come, from here small r can come, from here small y can come. What do you understand from that? The alleles of a character, my dear children, are independently assorting from the alleles of another character. It does not matter. Capital R will not say that I will only come with capital Y. They are assorting independently. It is going with capital Y, it is going with small y as well. Okay, so same thing happens over here as well, and that's how you get 4 gametes. These are the 4 gametes that you get on both the sides. Now you put them in a Punnett square and you cross them. So this is law of independent assortment, which clearly says that basically the alleles which are there, they are assorting independently. That is how you get so many different type of features. So now if I zoom this picture in, I have put all the 4 gametes here, similarly 4 gametes from the other parent there, and I have crossed them. When I cross them, since the gametes itself are different, the things that will be formed, the results that will be given, my dear children, that will also be different. So what is the different things that I find? 9 of them, they are like the parents, they are round and yellow. Okay, one is like the another parent, which is green and wrinkled, or wrinkled and green. But over here, 3 of them are wrinkled and yellow. Wrinkled was never with yellow, it was with green, but now it is with yellow. Similarly, green was never with what you call green was never with round, but over here it is with round. So these are the new features that you get because the alleles are independently assorting, my dear children. And there's an easy way to remember it. 9:3:3:1. So 9 of them, they are what? Round and yellow, that is the parental dominant type. And 1 of them is the parental recessive type, which is wrinkled and green, my dear children. Over here you have to find the new combination. So round was with yellow, now you make it with what? Green. Wrinkled was with green, now you make it with what? Yellow. So 3 plus 3, 6 of them are very new. Okay, 6 of them are going to be really new. Fine. So law of independent assortment, when 2 pairs of contrasting traits are combined in a cross, the segregation of one pair of characters is independent of the other pair. In simple terms, gene for different traits assort what? Independently. As simple as that, and then you have to mention this and you have to write all of it. This is the phenotypic, the ratio out there. And if at all you are asked, how many phenotypes are there? So you know, 1, 2, 3, 4. 4 different types, 4 phenotypes are there. How many genotypes are there? So not asked about, you do not have to count all of them, 16 of them. No, you have to find the unique number of them. How many unique number are there? 9. 9 unique genotypes are there. So many of things are repeated again and again. So 9 unique genotypes and 4 unique phenotypes. Remember that. Now, what is female heterogamety? Explain it with an example. Female heterogamety means females are going to produce 2 types of gametes. In the case of human beings, my dear children, we have XX in the case of females. So whatever female do, they will produce only 1 type of gametes, all of them having X chromosome. But in female heterogamety, they are going to produce 2 different type of gametes. And over here, you have to explain it with an example. So example is what? Birds. Birds are the example for this particular thing. So in the case of birds, it's ZW. So females are Z and W, and in the case of males, it is ZZ. So males anyway produce Z only, and females are going to produce 2 type of gametes. Crossing will be happening. 50% of them are going to be females, 50% of them are going to be male. You have to explain it with the help of the answer given below. Okay. Now, what are mutagens? Mutagens are the one that causes mutations. For example, UV radiation. UV radiation has a direct impact on our DNA. It can cause mutations out there. So that is known as mutagens. So mutagens are physical or chemical agents that causes what do you call changes or mutations in the DNA sequence. It can be X-rays, UV rays. That's why we say do not. In X-ray room, it is very clear that those of you who has a problem has to get inside that. Otherwise, you please move out. Why? Because it is not that great for our body. It's not that safe. Okay. Especially when it is done so many times. So yes, so give an example for it, write it properly, and that's all. Birds is something that you have to not miss. Now, given below a sequence of nucleotides in a particular mRNA, an amino acid coded by it, and then my dear children, over here this is a sequence and these are the amino acids coded by it. Methionine, phenylalanine, glutamine, leucine, valine, all of it. Write the properties of genetic code that can be deduced from it. So what do you understand? Do you find any commas, full stop, anything out there? No. So, no overlapping or what you call commaless. The codons are read continuously. The codes, my dear children, all of them, they are in 3 numbers. So, triplet codons are there. And the next thing that you can find is what? Basically, AUG is coding for methionine. UUU is for phenylalanine. UUC is for phenylalanine. It's not coding for anything else. So, it is known as unambiguous nature. Each codon which is there codes for a single amino acid, not all of them. Okay, and you also see one more very important thing. All this 1 and 2, they are coding for phenylalanine. This is known as, what do you call this feature as? Degenerate feature, where multiple codons are coding for the same amino acid. So this is also coding for phenylalanine, this is also coding for phenylalanine. So that is degenerate feature. And now at the end, my dear children, UAA is mentioned, and what do you call, parallelly you do not have any amino acid. Why? Because it's a stop codon. It stops the process of translation, terminator codon. So you have 3 stop codons: UAA, UGA, UAG, UGA, UAG and UA. So there are 3 things that you need to remember. Okay? And degenerate feature is also there. Do not forget to mention it. So I'll write it over here if at all by any chance you forget. Now, describe Griffith's experiment to show transformation in bacteria. So you have to talk about Griffith's experiment. What did he choose? He chose Streptococcus pneumoniae bacteriae. In that you have bacteria. In that you had S strain and R strain. S strain is what? Smooth. It has a smooth polysaccharide covering, my dear children, which makes it virulent. That means it causes disease. R strain is there, which is non-virulent strain, my dear children. And then he performed experiments. How did he do that? R strain does not cause any problem, okay? Now he put R strain in. First, let's take this. He put R strain into a mice and the mice lived. It does not have any problem. He put live S strain into a mice and the mice died. That was very sad thing, right? And now he took heat killed S strain. That means he did some changes in the S strain. He treated it with heat and then he put it in the mice. The mice didn't die. Unusually, the mice didn't die. Then probably something was getting damaged because of the heat, right? And now he did one more thing. He put a mixture of live R strain and heat killed S strain. Live R strain, which was not causing any trouble, and also heat killed S strain. When he put that into the mice miraculously died. How? Because probably something was getting transformed. And you know what he found? He didn't find. Later on, people found that S strain was there, live S strain. We didn't put live S strain, then where did live S strain come from? So probably the R strain which was there was getting transformed into S strain because something was getting transferred. So what is it? Conclusion was a transforming principle from the dead S strain converted the R strain into what? Virulent S strain. Something was getting transferred from here to here, transforming this into an S strain. Okay, and that would be the genetic material. That was the thing. And later on, Avery, MacLeod, McCarthy came into the picture and they gave all the things. So this is how it worked. You can go through it as well. Next thing, draw the schematic structure of transcription unit. So this is a transcription unit. There will be a template strand which is going to undergo transcription. It is going to be in 3' to 5' direction because the transcription happens in 5' to 3' direction. Always remember. So the new strand is going to be formed in 5' 3'. So this is template strand. This is just a coding strand. Over here on the 5' end of the coding strand, you have the promoter region onto which the RNA polymerase will come and bind. And then you have a terminator region. Know about the sigma factor and the rho factor. Sigma factor comes and attaches over here with the RNA polymerase for initiating the process of transcription, whereas rho factor comes over here and attaches at the terminator region because it is going to terminate, so that the RNA and the DNA can separate easily. That's why rho factor is needed over there. Now you are asked what is the difference between euchromatin and heterochromatin. So euchromatin, my dear children, when you know how the DNA is wrapped around in the nucleus. So DNA is wrapped around on some histone proteins. Okay, on some proteins which is rich in positive amino acids. Okay, such as lysine, arginine. And now the DNA is negatively charged, which is why it wraps around. It takes almost 8 histone proteins and to form what? A histone octamer. Now on this histone octamer, the DNA is wrapped, right? And you have another octamer, again the DNA is wrapped. Twice it is getting wrapped. So which is why you have a compact structure which is known as chromatin. Now chromatin can be of different types. It can be euchromatin, heterochromatin. Euchromatin is where the octamer that you see over here, they are far away from each other. They have some space. Whereas heterochromatin is basically where the octamer is really close. Okay, so over here they are going to stay in light. Here they are going to stay in dark because there is no space. Here they are going to undergo transcription because the DNA is there. Transcription can happen here. But here no transcription happens, my dear children. Okay, and this is loosely packed and this is tightly packed. Euchromatin is loosely packed and this is tightly packed. So these are the features that you need to remember. Cool? Chalo. State the Hardy-Weinberg equilibrium. Explain any 4 factors that affects the Hardy-Weinberg equilibrium. So Hardy-Weinberg equilibrium says that the allelic frequency of a population remains to be same. That means whatever characteristics is there, the frequency of it is going to be the same. There won't be any changes whatsoever. Do you think it can be like that? No. Why? Multiple reasons are there. There can be mutations happening, right? There can be mutations happening, certain group of organisms dying off, okay, certain others, coming into the picture. There are a lot of things that can happen, which is why Hardy-Weinberg principle or equilibrium principle was not considered to be exactly true. But anyways, it says that the allelic frequency in a population remains constant from generation to generation in the absence of evolutionary forces. In the absence of evolutionary forces. If at all evolutionary forces are not there, then my dear children, this is there. So mathematically expressed as p square plus 2pq plus q square is equals to 1, where p is what? The frequency of dominant allele, q is the frequency of recessive allele. Now factors affecting Hardy-Weinberg principle is what? All this thing, gene mutation, sudden changes can cause the DNA, and that is how new things, catalysis can arise, okay, that will be definitely changing the allelic frequency. Suppose capital T and small t is there, something else is also coming in the picture, then it can happen, right? Now gene flow or migration, for example, there are tall people in this area and short people in this area, but some short people came over here. Now you have alleles of the short people as well. So that is known as gene migration. Movement of individuals into or out of the population, changing their allelic frequency. Genetic drift. For example, I have some green moths over here, more in number rather than red moth. And then finally one, elephant came and stamped all the green moths. Now you just have the red moth. So again, allelic frequency is getting changed. That is known as genetic drift. This is going to be random changes. Genetic drift is a result of random changes, always resulted. Recombination, that may remember it, sorry. Recombination means new combinations are being formed. So that is there. It can happen because the chromosomes are lying very close to each other. Yeah. Natural selection favors certain alleles over the others. So naturally certain things are being favored over the others. You saw the example of the the white moth and the black moth, right? Dark moth and the white moth. So those are the things that you need to remember. These are the factors. Cool. Now, answer any one of the following questions. This is again, you have to answer any one out of the other options given below. You have 3 options, and out of that you just have to answer one. Double fertilization, unique event taking place in the flowering plants. Briefly describe the process. You have to mention 2 times fertilization happening. First time, one male gamete fuses with the egg cell, forming what? The zygote. This process is known as syngamy. What is happening? Egg cell is haploid, sperm is haploid, and the diploid zygote is being formed. Okay. The other male gamete fuses with what? The 2 polar nuclei. So 2 polar nuclei, individually they were haploid, one more haploid sperm came, or the male gamete came. Now you have triploid structure, which is known as primary endosperm nucleus. Primary endosperm nucleus. Since 3 nucleus are coming together, it's known as triple fusion. So syngamy plus triple fusion gives rise to what? Double fertilization. Now my brain is not working. Okay, hence 2 fusions occur in an embryo sac, a unique feature of the flowering plants, only the angiosperms. Now over here, arrange the following terms in the correct development sequence. Always make sure you're making a diagram for this particular thing, since it's a 5-marker question. Don't come out of the examination hall without making the diagram. Now, yes, you have to arrange all of them in the correct developmental sequence. So first thing, sporogenous tissue, my dear children, because they have all the microspore mother cells. The microspore mother cell or the pollen mother cell will be the second thing. So this is first, sporogenous tissue first, then pollen mother cell. Then pollen mother cell undergoes division, give rise to microspore tetrad. Each of them give rise to pollen grains, and the pollen grains have the gametes, the male gamete out there. Cool. Works? Fine. Now, next. Using the standard pedigree symbols construct. Okay, we will come to this question at the end. There's the last question. A health camp was organized in the village to conduct the routine checkups for the villagers. 5 patients with the following symptoms were recorded and preventive medicines were prescribed. Identify the diseases based on the given symptoms. High fever 39 to 40 degrees Celsius. Guess. What is it? High fever 39 degrees Celsius to 40 degrees Celsius. It would be typhoid. In typhoid you get fever spikes like that. Now chills and high fever reoccurring every 3 to 4 days. So every 3 to 4 days you're getting chills. Why are you getting chills? Because in their body it is affected, it is infected by plasmodium that causes malaria. And the RBC bursts open and releases some toxic things, which is why we get chills. Nasal congestion and discharge. Now people might think it's pneumonia. Is it pneumonia? No. Pneumonia affects the lower respiratory tract. It's common cold that affects the upper respiratory tract. Always remember. So this is common cold. Okay, don't do that mistake. Now blockage of intestine with internal bleeding. Blockage of intestine with internal bleeding is found in Escherichiasis roundworms. They affect the intestine. So this is known as Escherichiasis. Now this one, lips and nails turn greyish to bluish in color. It happens when you do not get enough oxygen. Now you have to tell me that answer in the, what do you call, E options answer in the comment section. So comment the answer right now if you know. I have already given you a clue. Okay, turns greyish to bluish in color when there is oxygen deficiency. So these are diseases. Know the symptoms of all the diseases, then only you will be able to answer a question like this, right? Okay, now we will come to this question. The last question of the day. My dear children, did you understand whatever I taught over here? Or it's like, oh my God, ma'am, I don't understand anything. If at all you understood, as I said, subscribe, share and like. Now, using standard pedigree symbols, construct a pedigree chart for inheritance of a X-linked disorder. That means it is on the X chromosome. It didn't mention X-linked recessive dominant, so you can go ahead. Given the normal male parent has 4 children, so they didn't mention about the female parent, okay? First of all, this type of questions you have to read it entirely. Out of the 4 children, only 1 male and 1 female carrier carries the gene of the disease. The affected son marries a normal woman. They get 3 children, out of which 1 is male and 1 is female. So they didn't tell anything else. First, we go line by line. So, normal male parent has 4 children. This is normal male. Out of 4 children, 1 male and 1 female carries the gene. That means you need to have a affected female. Okay, female should be there, a carrier female or a normal female. So this is the female that I have. Okay. Now, she can be just a carrier. Suppose it's a recessive disorder. You have to mention, okay, it's a recessive disorder. And this is a male with XY. Why I didn't make it XA over here? Because in that case, all the children would be affected, right? But here it's not mentioned like that. Out of the 4 children, now they are having 4 children. Okay, out of the 4 children, 1 male and 1 female carries the gene for the disease. 1 male and 1 female. So 4 are there. So I can make randomly. I have been told 1 male and 1 female is there. So I know about that. Rest of the things I can draw randomly. So I have made 2 females and 2 males. Okay. So the male carries the disease. So over here, my dear, in this condition, the males will be formed when this and this can combine, or when this and this can combine. Suppose here the normal X and the normal Y combine. And over here, XC and the Y combine, which is why the male would be affected. The male is the one that carries the gene. So we have to show this, that it is affected. He cannot be a carrier, he has the disease. Now, 1 female carries the gene. So female over here, if you make the female, my dear children, either it can be XX or it can be XXC. So over here, when I say it carries, let's make it like this. This is the carrier, but she is not affected. She is just a carrier. And the other one, my dear children, can be normal. Can be considered to be normal. Okay? Fine. Now, the affected son marries a normal man, normal woman. Normal woman means she cannot be a carrier. They get 3 children, out of which 1 is male and another is female. They didn't mention anything. So, marries a female, a normal female, and get 3 children, out of which 1 is male. So, I just know 1 is male and the other is female. I can make any random thing. So, I can make another female as well. Now, over here, when you make the male, you need to understand one very, very important thing. Is it going to be affected or not? That is the important question. Do you think would be affected? No. Because both the X chromosomes are normal and you have just Y chromosome. So, over here, this male will be fine. Female, my dear children, can be a carrier, cannot be a carrier also. Does not matter. So, yes, at least this is how you need to make the picture, make it properly and explain it, go step by step. Don't think very much, okay? Just go whatever is written over here, you have to think like that. This was not written initially. 1 male and 1 female carries the gene is written. So, don't color the female out there. Look, how is the not color the female out there. She's a carrier, she does not have the disease, so don't color it. Okay, so when you write, this is an additional thing, you don't have to write it there. When you make like this, it becomes all the more clearer. You can write it on the day of examination if it helps, okay? And then mention this is what X-linked recessive disorder. This is not dominant, this is recessive disorder. And recessive disorder, you find that it skips generation. Okay, so in this generation you didn't find it, but in this generation you're finding it. In this generation you're not finding it, the next generation you may find it. Okay, so that is a thing that happens. So disease skips generation through carrier female, showing it's an X-linked recessive disorder. You can make accordingly, whatever pedigree you're making, accordingly you can answer. Affected fathers pass the disorder to the daughters as carriers, but never to the son. So you can mention like that, or you can just say if a female is a carrier, okay, female is a carrier, the son would definitely get it. You can also mention that as well. That is what you understand from the pedigree, right? You can mention that the female is a carrier, the son may get the disease. Son will there are chances that he may get the disease, because it can be either like this or it can be either like this, right? If she is a carrier. So you can explain that. Cool. So that's all, my dear children. With that, I am ending this particular video. I hope this video have helped you in some ways. As I said, if at all you think it has, definitely let me know. With the help of your subscription rate, your likes, your shares and everything. And if you have any doubts, you can ask me in the comment section below. And as I said, do the homework question and let me know that as well. Wishing you all the best. You are going to absolutely ace the examination. Bye-bye. Take care.